There we go. Are we rolling? Great. Welcome everybody to the second edition of the Biostock Life Science Summit here at Medicon Village in Lund. My name is Jonas Söderström and I'm the CEO of Biostock. And we are extremely happy to see all of you here today. We have quite a packed program today. We will be listening to showcases from more than 20 companies today. So brace yourselves. We will also have a number of panel sessions with distinguished stakeholders in the life science industry. The theme for today is financing and investor insights, which is, of course, being in the life science industry, everyone knows that financing is the backbone for driving development and also driving innovation. So we at Biostock, we wanted to make this day about all the different stakeholders, the players in the industry that make innovation possible. So we have invited investors, financial experts, venture capital players, and of course then 20 plus companies who will be presenting to all of you today. And we also live broadcast this, and we will record it and publish it post event as well through Biostock's different media channels and also through our media partnerships in Sweden and in the US. So without further ado, I'm very pleased to be able to welcome the first speaker of today, which is Julie Silber of Immunovia. Welcome.
We're getting, aha. Okay, now I don't have to talk so loud. So we get a lot of questions from investors about this technology, ctDNA, which measures changes, it measures, God, I'm losing my English. It measures different tumor discretions into the blood system once cancer is detected in the body. And they measure our system against this, and it is actually very difficult to go this early on into detection with this type of technology. Our technology can detect changes in the system early on as far as stage 1 in various cancers, and I'll tell you a little bit more. Our first product is IMMray PanCan-d, and we have decided to go with pancreatic cancer because it is a very devastating issue. Right now, with current technologies, diagnosis is done in either stage 3 or stage 4, which doesn't give a whole lot of opportunity for the patient or for the clinician. The average median survival rate once detected is 4.6 months. When we move the detection rate down to stage 1, it has increased tremendously. Pancreatic cancer right now is growing as far as incidents are concerned. Right now, it is just actually in the last year or so has passed breast cancer in number of incidents, and it is actually growing, whereas colorectal cancer is being diagnosed earlier and being treated earlier. Lung and bronchitis is being treated earlier. Those two cancers are going to be going down. Pancreatic cancer is actually probably going to surpass colorectal cancer in the next coming years, and that's why we've chosen pancreatic cancer as our first indication. We are targeting 3 very high-risk groups, which comprise about 4.2 million people annually. The first group is the hereditary group, and this is an interesting group. These are made up of human beings that have 1 or 2 members of their family close to them that have had pancreatic cancer. The incidence for them of developing pancreatic cancer with 2 family members or more, and or some of the other hereditary issues is multiplied multiple times. It's not just a factor of 10, it's multiple. And these people are very, very worried, and they are willing to pay out of their pocket for a test right now. But there's also a group in there that's called the worried individuals, and they are the ones who maybe have 1 family member that have had pancreatic cancer. And once you've seen 1 family member die of pancreatic cancer, you want to solve this issue. And these numbers of people are quite large. We actually get phone calls and emails every day asking for access to the test. Second group is new onset of diabetes type 2 after age 50. It's an interesting thing because right now they're not sure if the new onset of diabetes is a result of pancreatic cancer or pancreatic cancer causes diabetes, but there's a huge risk, 8 to 10, risk of developing pancreatic cancer 1 to 3 years after diagnosis. So this is actually a target group that will need to be monitored for some time, up to 3 years, with multiple tests to see if their diabetes is a result of pancreatic cancer. The other group is the early symptoms group, and I'm not going to go through all of them, but these people go to the clinician, they get a diagnosis, they go to a specialist, and they go back and forth, and some of them end up with pancreatic cancer, and by the time they actually see the doctor and get the diagnosis, it's too late. So those are the 3 high-risk target groups. This is the addressable market, $4 billion. I'm going to skip forward because we have 10 minutes, and I'm not used to talking in just a 10-minute. Let me just talk a little bit. I'm going to talk about our optimization study results from this summer. So we did an optimization study, and I'm just going to go to this one during the time difference. We had quite extraordinary results. So we did a study comparing using our test with controls versus pancreatic cancer stages 1 and 2. And we used controls mostly from our key opinion leader network, and the results were an extraordinary 0.98, which is about 98% accuracy for these groups. And I'll go back a little bit. So we did healthy versus PDAC, we did diabetes versus PDAC, and we did symptomatic versus PDAC. So we tested everyone in all those 3 areas, and this is the results. And I like to stay here just a little bit because no one in the history of diagnosing pancreatic cancer has ever had this kind of results. Immunovia is the only company that has ever had this extraordinary results. So moving forward, what are the steps? Like I said, we successfully completed with a 95% accuracy for PDAC versus symptomatic diabetic and healthy controls. This was over the summer, and this is where we are now. So we just recently announced that we have commenced our study for the commercial test model, and what we'll be doing is using up to 1,200 samples collected from our key opinion leader network that will test the candidate commercial signature and fine-tune the algorithm. And after this, we'll lock it down and move on to the verification study and then the validation study. This study will read out at the end of this year, and then we'll move into this study, which will get Q2 results, and then Q3 results here, and then sales start. So we're really close, and we're really excited. I wanted to talk a little bit about our key opinion leader network, because this is really key, and this is also key in our new our pipeline products. So building this key opinion leader network is absolutely crucial. For one thing, getting the fresh samples for all our testing. 2, they are going to be our target customers. 3, in order to get reimbursement for future sales. They connect us to all the patient organizations, and then they also communicate on our behalf to a greater network. This is really important, and this is actually the most time-consuming thing in the whole process. And this is something that I think that we need to communicate better on, and I want to do that. So I am down to 1 minute. Quickly, this is our, let's see, I'm going to talk a little bit about rheumatoid arthritis. We just put out a press release on rheumatoid arthritis. The project is moving forward quite well. We have established relationships with new clinicians and key opinion leader networks in order to start getting the fresh samples in order to begin the testing. The next project that we're working on is lung cancer, which we also discussed in a press release a couple weeks ago. And this is 2 different projects. So one of the projects is a companion diagnostics project with a big pharma company. This one's moving along a little bit slower than anticipated, but it's moving along. The other is our own very important test, and that's going back to the foundation of Immunovia and doing early diagnosis. So this is also moving along quite well, and we have started establishing the key opinion leader network in this area, and we'll be getting samples in to start our testing. And, screen went off. And summary, in a nutshell, this is Immunovia. So we are very close to market, Q3 2020, and if you want more information, please come to our website. Thank you. Yeah.
Perfect. All right. Good morning, everybody. Now I don't have to scream. And do we have an image as well? Yes, we do. I'm here to present Spago Nanomedical in a nutshell, of course, since we have 10 minutes. Spago has developed a nanomedical platform which we can use to tailor nanoparticles to be used as either diagnostics or therapeutics. And we work with life-threatening diseases. We have chosen to start to focus on cancer, and I will get into our projects a little bit more as we go. First of all, just to say something about the technology that we have invented. We are tailoring a polymeric type of nanoparticles in a way so that they can accumulate passively in solid tumors. And the good thing about these particles is that we can tailor their size, which is critical to get enough material into the tumor. And they are also excellent chelators of metal ions, which means that we can bind metal ions that are useful, for instance, for MRI, which is our first project, SpagoPix, is dedicated towards. But also radioisotopes, so that we can use the particles for radionuclide therapy of solid tumors. We believe this is a unique platform. We have solid IP of this, and we intend to become the world leaders in the field of passive targeting to tumors. So starting with MRI, why is there a need for new MRI contrast agents, you may ask? This field is quite old. The agents that has been around for 25, 30 years now. But they're all rather unspecific. They are based on small molecules, and they are based on a metal called gadolinium. The unspecificity of these agents results in a very high percentage of false diagnosis. You see a lot of things in these images, especially when you're looking for tumors, and you don't really know what you're seeing. So this is a problem with today's use of MRI in solid tumors. The second problem is related to gadolinium, which is a metal that is not supposed to be in our bodies at all. And it accumulates in the brain, for instance. There's a huge debate going on in the field what this means clinically. But what it has meant or do mean already is that patients are getting worried, physicians are getting worried, and authorities are getting worried about this. There's also the fact that these agents disappear very quickly from the body. They are excreted within a few minutes, which means the imaging time you get is very inflexible. With Spago PICs, it's quite the opposite, actually. Spago PICs accumulates in the tumor and lightens the tumor, whereas the background remains unenlightened. This tumor selectivity can help reduce the number of false positives, we believe. It's also based on manganese, which is an essential element in our bodies. We get it with food and water every day, and there is a mechanism in the body to take care of excess manganese. So it's perceived to be less harmless than gadolinium. With the particles, we also can achieve a longer retention time in the body, allowing for more flexible imaging. So taken together, this makes Spago PICs a potential new agent to improve MRI, and especially MRI precision in solid tumors. Now, so how do we use this? We have chosen to focus initially on breast cancer. And the reason for that is that we have preclinical data that is solid and supports this. There's also strong support in the literature for use of these types of agents in breast cancer. And not the least, MRI is already an established technology in breast cancer. It's already in use for the characterization of these patients. And of course, breast cancer is a huge indication, as you all know. And a large fraction of patients already undergo MRI on an annual basis, especially in the US. There's also a medical opinion for the increased use of MRI in these patients. And that is because MRI is a really good method. It's better than mammography, it's better than ultrasound for spatial characterization and aggressiveness assessment and so on for breast cancer. So, and a premium, a specialized product in this field is considered to give a premium pricing as well. So with that, Spago PICs is targeting a significant market and has also, not to forget, the potential to expand into other solid tumors. So we believe this product can be of great use for the whole MRI field, actually. Now, just a few words about our second product, which is a bit earlier in development, but targets an even bigger market. TumorAd is based on the same technology. It's based on our unique nanoparticle platform. It uses a radioisotope, and it's given systemically and by passive targeting accumulates in solid tumors. That means we can treat solid tumors from the inside with a radioactive agent. And we know radioactivity works, right? It's been around for a long time. It's still one of the cornerstones of cancer therapy. The trick here is to get enough of it into the tumors while saving the rest of the body, of course. And this is exactly our speciality within Spago. Also, with the current radionuclide therapies, there is sometimes problems with the logistics because radioactivity needs to be handled, it needs to be coupled to the active carrier and so on. In our case, it's much easier. It's a matter of having particles in one tube, having the isotope in another tube, mix them together at the hospital, shake, and then you're done. So this is, sometimes people forget that the actual logistics at the clinic is really important if a product will succeed or not. There are some recent benchmarks which is very favorable in this field. Xofigo was approved for skeletal metastasis coming from prostate cancer in 2013. Its predicted peak sales is now $1.2 billion. And a similar product, Lutathera, which is based on an antibody targeted towards somatostatin and is used for a niche indication within neuroendocrine tumors, was recently approved and is now predicted peak sales of $2 billion. So we are targeting huge markets here. And again, Tumorad holds potential for broad application in solid tumors. Our particles accumulate in many different types of solid tumors, and especially aggressive ones, which means that we can actually target a growing therapeutic segments, and there is a need for new therapies, as you all know. So just to sum up, and this here is the setup right now at Uppsala University Hospital for our clinical trial with Spago PICS in breast cancer. The study was started just a few weeks ago, and we have the first patients in, and all looks good so far. With Tumorad, as I said, we are working on a lead candidate, and we are making a lot of progress there as well. We have allocated more resources over the last year to this project. So the next milestones will be first interim data from the trial with Spago PICS, but also declaration of the lead candidate in the Tumorad project. Please don't hesitate to contact me with questions, and thank you for your attention.
Thank you so much. And next up, we have the CEO of Aptahem, Mikael Lindstam.
Thank you very much. I'm sitting almost sleeping, despite the fantastic.
Already boring you.
I've been up all night trying to prepare this presentation,
well,
because we have 10 minutes.
Well, good luck.
Instead of 10 hours, so I will do my best in these 10 minutes to do something about it. Well, I'm Mikael Lindstam. Good morning, everyone. I'm the CEO of Aptahem. And I will take you through Aptahem briefly, how we started, where we are, and where we're going. But most formally, I would like to talk about Apta1, our lead candidate, and its unique abilities to be a potential candidate against sepsis. So, we were founded 2014, made a public appearance 2015, and we are a biotech company. So we are addressing life-threatening conditions where inflammation and coagulation is a factor. And that means we have a group of aptamer drug candidates with very unique abilities. We are sited in Malmö, and we have been there since 2014, as I said. So, a little outlook what happened. I will not go through everything. You can talk to the team, which is here afterwards at the break. But anyway, 2014 founded, and we did it on anticoagulants. And anticoagulants showed fantastic abilities with Apta1. And down the road, we found more and more information that also pointed out that it was an anti-inflammatory agent. And that meant that, we have a fantastic combination. So we put it in the worst model we could think of, namely a sepsis-like model. And the outcome was shockingly good. Super survival. So here we are at something. So 2017, the company made a strategic decision to change to focus on sepsis. So from there on, we have been going with new fantastic results to support Aptawan's potential. And also, we built a great team. We had made good collaborations, and now we are here. And recently we released a big report, 200 pages, that confirms and fundaments everything we have seen so far, and also new, interesting findings. Let's see. This is wrong. Oh, there. Sepsis. I already said sepsis, perhaps. This is an awful condition to be in. The death rate is very high. There's a lot of people getting struck by this, indirect by different sick conditions. It could be cancer, it could be pneumonia, whatever. This is an indirect effect mostly, and usually bacterial struck. Many dies from this, and there's no cure. So there's 3 big, very strong points of the unmet need, and it's a global, very big unmet need. So, company rethought this whole idea about how could we possibly use Aptawan as a potential drug candidate against sepsis. Yes, we did that 2017, and since then we've been going on. And on the right side, you can see how many incidents in Europe we have, 1.2 million, close to half die. This is serious stuff. And 30 to 45% actually doesn't get the correct cure that can give you an aftermath. If you survive sepsis, you can be in big trouble. The dialysis, amputations, and so forth. And it has a big, big burden on the socioeconomic system. Family members, colleague at the work, and also the aftercare. It costs us enormous money, except also the personal suffering. I just want to say something here now, and that is Aptawan is not the one target candidate against sepsis. It's a multi-target candidate, and I will go soon into this why it is like this. But most efforts in these areas have been on one target candidates, and so you will see here soon. There. First, a little about Aptawan. It's an RNA aptamer. And what does it mean? Yeah, small fragments of RNA has built into a specific target protein and built a very strong specific structure that binds very strongly to the target proteins. And generally, aptamers are very non-invasive. So they just go through the body, do nothing if there's nothing to attack, which has to do with the target proteins that it's been modeled after and tailored after. And so far, we haven't seen any big side effects or anything out of this. And this is just an example how Aptawan, as you see, quite an advanced structure, many active sites because it's multitasking. And it just specifically binds in very strongly. So, before we talk about how Aptawan works on the sepsis case, or the potential sepsis case for the Aptawan, is how does sepsis work? And we try to very briefly go through what's happening. We see 2 stages here. First stage is in coagulation and fibrinolysis. These are the 2 systems that regulate the coagulation. You need to make clottings in the system, coagules, and then you need to declot them. And that is a system going around all the time. If that gets struck out of balance, you have a problem. And the body will react following to this unbalance that we have. And sooner or later, that will lead to an inflammatory response in the body, which is stage 2. And here you see all the stages. Stage 1, sepsis in the middle, severe sepsis, and when you come into the inflammatory stage, when it runs out of control of the body, it gets dysregulated, you have a septic shock. And that means you are in big, big trouble. And why do we get into septic shock? Yeah, well, when you get the inflammatory response on the vascular wall, where there are coagulants sitting, then you have an inflammatory response, and that leads to tissue breakdown, and tissue breakdown breaks down the vascular, and the vascular starts leaking. Hence, what will happen? Yeah, blood pressure goes down. And at a certain level of lowered blood pressure means you go into shock. And shock means that you have not efficient oxygenation of the tissue and the organs. You have an organ failure, and you die, sadly enough. So you see here, septic shock is the last stage before you actually pass, if you can't take it. And DIC is just an abundance of coagulation clots in the system, and that can lead to a massive stroke. You can die of that too, if you don't die from bleeding or the shock itself. and death we talk about. So I talked about one target. Here is plenty of one targets that failed, both in the stage 1 and stage 2. So remember, we are a multi-target, and here we have the solution, at least as we see it, the potential solution to address sepsis as a very, very life-threatening condition. We take care about the coagulation. We counterbalance the system. So it can stop coagulation. It can help dissolve the clots. In the inflammatory part, we can press down pro-inflammatory cytokines. We also have a stimulation of the body's own system, which is a fantastic feature. So Aptahem does something magic. Of course, we know a lot about how Aptahem works in this system. But I just want to say that if you have questions, the CSO is here, sitting here in front, can take questions later. And of course, read on our homepage and other things. And we hope to come back with a longer presentation, not 10 hours, but certainly longer than 10 to give a better insight into our technology. But what this means, we have a tissue repair, we protect the organs, we have a maintained blood pressure, no shock, potential survival. Fantastic. So, yeah, we have some new insights. We just recently released a report on the NHPs that we've done, and it's a massive report. And it contains a lot of facts. And I would say that it confirms a lot of the things we've seen so far. It shows new indications. And we just strengthen our beliefs in that this is a very potential drug candidate. And some new things that we see is actually the potential for the body's natural system to protect or repair tissue damages. And also we have this pro-fibrinolytic effect. This is a fantastic feature to be able to balance the body's coagulation system so it doesn't run away to clots or anything like I just said. Anyway, we also see another thing that may play a role in autoimmune conditions, and is that we can clear or we can stimulate the body's particular proteins to actually clear dead cells, apoptosis, that the body is working with. It's a very interesting feature. That may be a third patent family in the making, but we will see. We will come back to that when we have that if and so. Patents I talk about, we have 2 patent families. The first patent family is about the structures. We have 3 structures, whereof 1 is the lead candidate we're working with, Aptahem. And then we have the second patent family, which is an in-house invention. Inventor sitting here, you can talk to her afterwards. It covers a lot of therapeutic use, and it was published this spring. So you can go in and read this publication, of course, and take more in-depth information from that. And this is very important for the company, of course. This is a very high-valued position to have patents. That is do or die in the process of finding a partner. Anyway, third family may be on, but we have a very long protection as we speak. So we are very happy about that, and we try to prolong that with new patents, of course. So looking at this market, yeah, we need a market, and I just mentioned partners. Partners is needed to take it all the way to market. It costs billions of dollars to get something on the market. So we need to have a very good valuation here and potential market. And we have, look at the figures here. This is just an example where we address just immuno-related drugs in the treatment of sepsis. Not any antibiotics or systems that get up the pressure to keep oxygen in the system. No, so these figures could be higher. And look at this, 32 in Europe and 11 in US when it comes to immuno-related drugs. So there is something for us to catch more if we have the success we want to have. The team, fantastic team. Without the team, I would be nothing. I can say that immediately. Amazing team we built during the years. Very seasoned people, very skilled. Fantastic to work with them. And not everyone is here, so. And to wind up, we just have a warrant going on, and we had a strike price announced today, actually, and it will be exercised now from tomorrow, Friday, yes, to the 11th of November. And I hope you join us in that. And just to round off totally, we have things going on. We're waiting for a big batch of Aptahem that will kick in the GLP talks that we need to finalize the total safety talk studies to go into clinic. Thank you very much.
Thank you so much. That was really interesting.
Okay, thank you.
And with that, we've ended our first session, and you can all go out and have some tea and coffee, I think. And we'll be back at 10:20.
Was it exciting?
Oh, welcome back from the break, everyone. I hope you enjoyed refreshments and a bit of mingling outside. It's nice to see that so many of you have still decided to be with us. We're going to go to the next point in the program for today. We're going to have a panel session. And the subject of this panel session is, of course, investor insights. And we call it In Search of the Next Nordic Unicorn. And with me, I have 4 lovely, distinguished guests. It's Björn Odlander from Healthcap, Professor Carl Borrebäck, Lund University, and a few more things. We have Ingrid Teigland-Akai from Hadean Ventures, and we also have Johan Körel from Lundbeckfonden Ventures. I think we should start with giving them all a warm hand. So, I would like to start with a short introduction round with all of you. So, would you like to start, Björn? Who are you and what do you do?
Good morning. So my name is Björn Odlander. I'm the founding partner and still the managing partner of Healthcap, which is a dedicated life science venture capital fund based in Stockholm, but investing internationally. Closer like that. So by training, I'm a medical doctor and a PhD in medical chemistry from the Karolinska Institute. And by heart, I think I still am a medical researcher. That's what I identify myself with. And being in venture capital in medicine, it's a wonderful opportunity to be able to support interesting science that makes a change, makes a difference for patients. So Healthcap, in short, is we are currently investing out of our 8th fund. We have invested in more than 100 companies since inception in 1996. We have taken 43 companies public in various stock markets. And our companies, which is the thing I'm most proud about, have brought more than 25 pharmaceutical products to the market, in addition to about 50 or 60 medtech products. So that's me and Healthcap.
Impressive. Thank you very much. Carl?
Yes, Carl Borrebäck. I'm a professor focusing on cancer immunology at Lund University. Previously, I was the vice president of Lund University for innovation and industrial collaboration. Started 6 companies, spun out of the research we've been doing. I'm active in 4 of them, all listed. Raised 1 or 2 billion in venture cap from, not from you actually.
We'll change that.
So I'm on the receiving side, so have some experience in when it's about financing startup companies.
Great. Ingrid?
Yeah, thank you. I'm Ingrid Teigland-Akai, managing partner and founder of Hadean Ventures, which is a fairly new life science venture fund. Newest of the ones up here, at least. We launched the first fund of Hadean Ventures end of 2017. We are based in Oslo and in Stockholm, and we have a European mandate to invest in life science startups. But we have a particular focus on the Nordic region, where we see lots of very interesting investment opportunities, like we see here today, many examples of that. We are quite agnostic. We invest across technologies, pharma, biotech, medtech, diagnostics. So far, we invested into 4 companies from our new fund. One of them is based here in Lund. 3 of them are in the Nordics. So the Hadean Ventures team is a spin-out from a London-based venture capital fund. So I've been in venture capital for the last 10 years, and before that, I was also a medical doctor. Like Björn, worked in internal medicine and gastrointestinal surgery.
Thank you.
And Johan Körel. I'm a physical chemist, and I have a PhD from LTH. So I'm very happy to be right here, right now. I work for Lundbeckfonden Ventures, which is a Danish venture capital fund. And I've been there since its inception 10 years ago. And Lundbeckfonden typically invests in private companies, and then, if needed, follow them into the public arena. We have to date made 32 investments in the US, about half, and the other half in Europe. Unfortunately, only 4 of these investments have been in Scandinavia, and only 1 in Sweden, which is a shame. So I hope to hear more from you today.
Thank you very much. You all have fairly impressive backgrounds, I would say. Looking at the first talking point of the day, early stage financing, we'd like to know a bit about the entrepreneur's perspective versus the venture capitalist perspective. And I would like to start actually with you, Carl. Being an entrepreneur, having been at the helm of several companies that they have funded and also taken public, what would you say have been the biggest key learning for you when it has to do with early financing, to finding early financing for your companies, if there is one?
No, there is not one. I mean, this is really a complex question. I can talk for hours with, about this, but if you look more general, it depends very much on the stage of the management, stage of the company. One problem we have in academia is we cannot pay for our own patent applications, so we tend to start companies a bit early, and then you have sort of a challenge to approach venture capital at that stage. So what we try to do at least in the most recent companies is to avoid venture capital in the early stage, because you get into discussion about dilution, about market cap, and so on and so forth. And at least in the beginning, we are on opposite sides. Still be friends, but we are looking at this on opposite sides. So we've been trying to use private equity as much as possible for the round A. Later on, of course, venture capital is absolutely needed, because then we talk about much larger sums than a few million. And as I said in the beginning, we probably raised 1 or 2 billion in venture cap. And I probably shouldn't say that we should avoid venture cap in the beginning, but I think there is a point, because I've seen horrible, not from these guys obviously, but examples where the founder has been very rapidly diluted, and then you lose the whole idea of having a startup company coming from someone. So it's a balance, and it should be a win-win situation. But I think today there's so much capital in Sweden, particularly in Sweden, private capital. So it's absolutely doable to start companies like that.
Björn, having then, like you said, been part of funding quite a few companies and startups and going a long way with them, what's your perspective as a venture capitalist and to what Carl is saying here around the early stage financing?
It's not uncommon to hear these remarks from entrepreneurs, and it's sad. But it's all about choosing your partners, really. And first, a couple of sort of other facts about Healthcap is that we in the majority of the companies we invest in are companies we found ourselves together with entrepreneurs. So to come to us later is an uphill struggle, because there's so much we need to fix when there have been business angels in there, and it's such a sort of potential conflicts built into companies. So it really raises the hurdle for at least getting us in. We want to be really early into the companies. Then I think we have a motto, it's work hard and be nice to people. And one of our most important assets is the network we call Health Chaps. That's people who we've been working with, repeat entrepreneurs, repeat management, repeat board members, etc. And you need to behave in order to earn these people's confidence. And you need to earn it in order to be able to make more than 100 investments and been around for 25 years. So I think that speaks for itself when it comes to Healthcap. And most professional venture capital organizations that have been around for a decade or more. So, but it's, as I said initially, it's not uncommon to hear this from entrepreneurs, but you need to choose your financing partner. That's the simple answer to the question.
Carl, you want to?
Yeah, comment. I mean, Healthcap is unique in that sense that they would like to be part from the start, but not everyone is doing that. So from that point of view, I mean, then you can because what you can use venture cap is of course the network, and what they know is business development and taking it to the market. That's absolutely great. But if you not mentioning any other names, but there are a number of venture cap that are not working like you are, and that's when you should have a bit cautious approach.
So there could be a built-in conflict between the interest of the entrepreneur to have their idea mature a bit before you take in external bigger capital, and then also then from the venture capitalist perspective to see that you have done the right things from the beginning. Is that correct? I would like to ask you, Ingrid, how do you work? How do you approach this problem?
Yeah, so we do a little bit of both. So we can also, like Healthcap, be part of creating a company and invest into companies that already have angel investors on board, which can be a challenge for sure because of these alignments of interest that you talked about. I think from a sort of founder manager perspective, as Björn says, if you choose the right financing partner, that really shouldn't be a problem because we need to have our people on board motivated and incentivized to work for the company. And we want our founders and the management of the company to have enough upside to have that incentive, and partly because we're nice people, but also partially because that works for the business model. So that's very important for us. In terms of where you find the financing, I think it very much depends on the environment, and I completely agree with you. Right now in Sweden and also other places in the Nordics, there are lots of capitals available. From angels who think this is a super exciting space, but that doesn't always last. It goes up and down in popularity, and when you look at venture capital investors who are specialized in this space, I mean, it's our job to invest in this space. So we're going to keep interested in investing into startups. So yeah, it makes sense to look for financing from various spaces, but keep an eye on the specialist venture funds as well, and don't push them away.
Because you are all nice people. So, Johan, are you a nice guy as well from Lundbeckfonden Ventures?
I think you have to ask the CEOs of the companies I've been on the board of, but I think they'll say yes. I mean, like Björn, we had a maybe less polite motto when we started out, because we are all ex-CEOs and we've all raised money from VCs, old school VCs. So we decided that our motto should be, "Don't be assholes." But coming back to this theme of what we're talking about here, you get the owners that you deserve. You should really think about that, okay? So I think we all have a place here, but it's also right that we don't necessarily have the same drivers. So if you are after making money, then VCs are after making money, and they will make sure that if you're important for the company, you will also make money, because VCs have a long-term game. We want you to come back with your next company if we've been successful together. But if your goal is to help humanity, which is also VC's goals, then there might be different drivers. And if you have a purely financial investor who doesn't want to help humanity, and then you have a problem, right? So you have to really think, if you are in a luxurious position to be able to choose your owners, you have to think about what owner you invite into your group.
I would like to, you said something there if you are important to the company. That sort of transitions into another talking point that was investor activism versus active ownership. And when you say that, if you're important to the company, that sounds a bit like if you're not, you're going to be ousted when we get in there. Is that how it works?
Thank you for putting me on the spot. Again, comes back to, you know, how do you treat people? You have to treat people with respect. But if you only have X million Swedish crowns to do something, you have to focus that on what needs to be done to get to the next stage, keeping in mind that you want people to come back to you with their ideas. So I'm not saying that you hire and fire people, but you need to have the right staff in the company. And when it comes to the ownership circle, which it sounds like Carl is maybe more concerned about, you have to take care of the owners so that the owners are treated fairly and that they come back.
Carl, how about that? You mentioned earlier the dilution problem, that if you get into the big capital too soon, you, as an owner, a founder, you're diluted. But what about this other part, where helping out with creating a team and so on? Is that something that the VCs can help with, or you feel like you can do it yourself?
Just a comment before.
Yeah.
I mean, we all been sort of due diligenced in our startup companies, but I think it's very wise to also do due diligence on the venture cap, because they're so different. I can give you as many examples, but I won't. But just here, there are different approaches, and in general, they're very much different. And I think it's good to know who you actually get into bed with, relating to how long can we keep the founder. I mean, I said sometimes it's the first thing we have to get rid of is the CEO as a founder, or founder as a CEO, because then normally they do many good things, but they're not really business people. So I mean, coming then back to the question, building the team is absolutely the most critical thing in my mind. And the old saying is, it's better to have a good team and a poor idea than vice versa. I think it holds true. The problem is that we don't have so many good business developers in Scandinavia. It's really difficult to find them. And I think that one of the first people you should hire is a business developer. And from that point of view, you can really have a lot of help from the VC companies, since they have a network that most people don't.
That's one thing that you mentioned, Björn. You have a certain name for your network as well. What was it called again?
It's called Health Chaps, so it's a bit of a joke with words, of course, but it's for real, and we really believe in maintaining long-term relationships with people. So we nurture this network, invite them to meetings, listen to when they come with new ideas, etc. Offer people to recycle on boards, etc. And also CEOs, because sometimes, as Carl alluded to here, there is a CEO cycle. The same person who starts a company might not be the one who takes it public or commercializes a product. And that means that you need to have a dialogue with these people. And best for us is if a startup CEO steps off and starts another company, which we can fund. So it's about creating sort of a perpetuum mobile.
Mm-hmm. Ingrid, yeah.
Yeah, just a comment to what you said about, finding the right people, and it's not that many with a lot of experience in Scandinavia. I think, I'm from Norway. Norway is the smallest country of the Nordic ones, and the rest of the countries are not very big either. So usually the way we think about it, we have to stop thinking about ourselves as from Norway or from Sweden or from the Nordics. We have to think about it as European and use that as a market for, looking for capital and looking for talent for our companies. So I think it's important to sort of broaden our horizon a bit when it comes to our industry.
Yeah. We are coming towards the end. We have 5 minutes left or something. I had a few more talking points, but it's very interesting to hear your different perspectives. So I'd like to talk a little bit about what trends are you watching closely at this point? If you look at, like you said, we're Scandinavia, we're small countries, but we have a European or global perspective. So which trends are you monitoring right now, Björn?
You know, there's so much to monitor all the time. But we are living in a world of constant change, right? So you need to be open-minded and on the lookout all the time. If you want some specific trends now, I would be worried for where the financial markets are going. And the situation now is very similar to 1999, to be frank. And as I don't remember who said it, maybe it was Ingrid, that there's a lot of capital floating around out there right now. That's very volatile capital, and it can be gone in a moment. And the other trend is, of course, demographics. That's a much longer, and that's a trend. We have a growing population, and we have increasing age of the population. So the need for healthcare and for good treatment is increasing. And if you combine that with increased global wealth, which is also a very strong trend, that means that life sciences as a market is super interesting. And the third trend, which I think is here to stay as well, is the degree of innovation. Innovativity is increasing both in pace and in depth. It means that you can make much more educated bets on biology. So those are my trends.
Johan, which trends are you watching?
So then I have to pick up on the science. I think the microbiome area is fascinating. That, we've discovered a new organ, not inside our body, but sort of inside our body, because it's in the gut, right, which is the outside. I think that's very fascinating. I think what's happening in gene and cell therapy is very fascinating. But I have to say, and this is a lesson Björn taught me very early on in my investing career, there's a difference between being an asset investor and an equity investor. And Lundbeckfonden is an asset investor. So Lundbeckfonden invests in projects that could be products. And that's how they think, or we think that we can make money. Now, if you invest in equity, it doesn't really matter if you have a product or not. You have to have the idea that there could be a product and you can make a lot of money. So if you take the example of gene therapy, where we still have to see proper controlled clinical trials that are successful when it comes to gene editing, not gene therapy in itself, but gene editing. So we're very early in an asset perspective, but there are already successful companies that people have made a lot of money on that are public. So, if you're an equity investor, it's a fantastic field. If you're an asset investor, we still want to know where it's going.
And Ingrid, from the Norwegian perspective, which trends are you?
Yeah, well, we see ourselves as European.
Yeah.
So, as a particular trend that I can mention that the other ones have not mentioned yet is digital health, which is taking like a large and increasing part of our deal flow. Both sort of pure software plays and also technologies have an impact on previously more traditional life science firms. That is definitely a big trend that we're seeing.
And Carl, being very active in the space and very, connected here at the Medicon Village in Lund, is there any specific trends that you're picking up from your point of view?
Well, the most specific, of course, we keep to ourselves because we're going to start the next company. But I agree. The microbiome is super interesting as an immunologist. I look at the microbe as the back office, and they tell the ninja warriors of the immune system, the T cells, what to do. This is a completely new concept. And we know that some of the immuno-oncology therapies doesn't work in one patient with one microbiome, but work in the other patient with a different microbiome. This is just the beginning. The other thing is what you mentioned, but I think I would go a bit more specific and say digital companion instead of conventional companion for developing drugs. You can now have digital companions, which is coming more and more. And I think e-health as such is a bit, you have to be specific because it's everything and nothing. But in particular then in our own last company we started, most recent, we do digital companion with some large pharmaceutical companies regarding monitoring chronic pain, for example.
I'm going to have to say that you will be welcome next year to present that company here on stage. I would like to say thank you very much, everyone. The time is up for this panel. It was a pleasure having you. And I sense that the discussions might go on in the break later.
What do you mean?
Yeah, no, I'm happy to see you all smiling. Thank you very much. A big hand. So, yeah, come on. Next speaker on stage is Björn Dahle from Cereno Scientific.
And we are 2 minutes on my time.
Yeah, we'll make up for that.
Okay, thank you. Sure. Thank you, and yes, I'm going to pick up where the others ended. They talked about genetic therapy, genetic gene therapy. That I'm going to talk about epigenetics a little. So I think that's exciting, somewhere in between. I'm CMO of Cereno Scientific. I need this one, yes. And I'm going to tell you a little about what we do. Let's see, where do we have to point it to? Nothing happens when maybe I have to turn it around. No? Yes, okay. We are a company in the clinical stage, focused on cardiovascular disease. We have 2 products for the moment, CS1 and CS014. CS1 is a reformulation of VPA, a dual-action drug that's aimed for thrombosis, biggest killer in the world. We are also assessing the possibilities of expanding our scope in new areas like fibrosis, inflammation, and hypertension. We started 2012. Our journey started with small capital from Immunovia, went on the spotlight market in 2016 and concluded phase 1 2018, and 2019 have been a very, very fructious year. We have had a portfolio expansion. We have created a very exciting scientific advisory board, a phase 1 publication, and having approval for phase 2 trial. We are in the middle of where the excitement is, in the middle of AstraZeneca, the BioVenture Hub, which, according to Medicon Group, is one of the most novel incubators in the world today. What do we do? What do we affect in the body? We restore the endogenous fibrinolytic system to be activated when and where needed. In a healthy vessel, fibrinolytic capacity takes care of blood clots which are threatening to occlude the vessel. So patients with risk factors have a deficiency here, and they risk the occlusion, causing a myocardial infarction or a stroke or something in that area. We restore the capacity of the system to prevent that occlusion. And how do we do that? We have a new formulation of VPA, a drug that has been around for almost 50 years. And we have done an delayed immediate release formulation, which is patented. We can restore the capacity for TPA and block the activity of PI to kind of take care of the clot formation. And that happens only when and where needed. Not that we put the whole body in a near bleeding state. We just the system is activated when it's needed. And we have in vitro studies, preclinical models, proof of concept, human physiological studies, epidemiological studies that supports our concept with VPA, and a phase 1 study that has been concluded. So this is the proof of concept in animals. It is a mouse model where you create a thrombus formation with pre-treatment with VPA for 96 hours, and we could reduce the size of the thrombus formation without causing bleedings. In man, we have the forearm model where we can see that both in healthy individuals and in patients with atherosclerotic disease, coronary artery disease, that you can both increase the level of TPA when needed, and you can reduce the plasma pi, both indicators of a thrombus reduction. So the epidemiology is quite strong. VPA used in the whole world for epilepsy have shown that these patients in various countries, actually Denmark, Ireland, and in the UK, show reduction of myocardial infarction and stroke in the range from 25 to 50%. So I think that we are onto something there. The phase 1 study showed that with our formulation, which is given in the evening, gives a peak in the morning hours when pi is the highest in the body. We showed a very good safety and tolerability, a significant reduction of the pi levels, no bleedings, and the targeted pharmacokinetic profile we wanted. That was published this year. So when we go into the thrombosis market, of course, we are up to a very tough competition, especially the NOACs, which are very tough to compete with. But I think with our profile, having antithrombotic efficacy with minimal or no risk of bleeding, and also some possibilities opening up in organ protection with antifibrotic, anti-inflammatory effects, and also reducing pressure in both the pulmonary and the systemic circulation. And you all know the thrombosis market is a huge one. If we just go for one of the NOACs, we can project that we are into a billion-dollar market if we are successful. We have a very strong team with a long experience, both in big pharma and small pharma. Sten Sørensen is our chief executive officer. He has a long experience from Astra and other companies. I'm the CMO, and we have also strong financial and business, and we just tied a new business analyst to our team, Paula Salmer-Sandrak, very efficient. So we have a very strong team, and many of the founders of the company are still in possession of quite a big stock in the company, and the top 10 shareholders, which includes all the founders, have about 40% of the company still. We are also very lucky to have tied a very strong advisory board with Professor Bertrand Pitters, the chairman from Michigan, Ann Arbor, and Gordon Williams, professor at Harvard, together with Deepak Pat, which is the key opinion leader in thrombosis in the world, Fayez Sanad, and also Gunnar Olsson, which has a long track record from AstraZeneca. And they said at the meeting just recently, we should further explore the use of VPA-CS1 for epigenetic modulation in cardiovascular disease associated with fibrotic inflammation and pressure increase, both systemically and pulmonary. And we have taken up on that, and we aim to, during the autumn, broaden our scope. For the moment, our portfolio CS1 is entering phase 2 in orthopedic surgery, orthopedic knee surgery to prevent thrombosis. CS014 is still in preclinical. In thrombosis, we aim for doing the study in knee surgery, deep venous thrombosis, but later going to stroke prevention in atrial fibrillation. What's upcoming in our company? We need to make strategic decisions about our broadened scope in epigenetic modulation, in fibrosis, inflammation, and systemic and pulmonary pressure. What kind of indication could that lead to? We need to have all the approvals for the phase 2 study. We have already gotten some, and we need to scale up our production of goods and start a phase 2 study. So what's our value proposition? We have a product portfolio which includes the lead candidate CS1 and a runner-up CS014. This is a dual antithrombotic with a minimal risk of bleeding and is based on a novel formulation of VPA, has been approved for phase 2 study in preventing thrombosis in orthopedic knee surgery, and CS014 is currently in preclinical. The possibilities to expand our scope is really exciting with the epigenetic modulation in cardiovascular disease. In addition to that, thrombotic effect also show antifibrotic, anti-inflammatory properties and pressure lowering in the pulmonary and systemic circulation. Thank you very much for your attention. Yes.
Thank you so much.
Okay.
And you actually finished before time.
Yes.
We like that. Right, so next up, the CEO of Endovab, Stefan Sova.
Okay.
Thank you. So I'm Stefan Sova. I'm going to tell you about this exciting investment opportunity. There's a strong trend to go from open surgery to minimal invasive surgery. And there are many techniques. When you go through the blood vessels, it's called endovascular, and this is what this company works with. The procedures are done under X-ray, and the products are called stents, balloons, catheters, and guide wires. And there are 50 million guide wires used yearly in the world, and the first product is the guide wire fixator. The innovation is the first product in the world with temporary arterial anchoring with maintaining blood flow. So now this is a private health company. We develop and sell products for temporary arterial anchoring. 3 patents in 3 clinical areas, and company certified, ISO certified. We have a CE marked product, commercially available, clinical proven, and clinical evidence published. We are seeking funding in 2 stages. First, to verify the market and grow sales, and to expand the market and develop next generation products. There are 3 clinical areas where we have patents: aneurysms, peripheral artery disease, and precision chemotherapy. And we have a product aimed for each. The guide wire fixator to enable also county hospitals to do complicated aneurysms repair. In the peripheral artery disease, the filter fixator will protect from embolism, and the precision chemistry, the microcatheter fixator, will enable local direct drug delivery. More details on coming slides. This is the element. Key element is made of a memory metal, and at body temperature it regains its predetermined shape and presses against the vessel wall. When you don't need it anymore, you can retract it into the catheter. There are 3 products in plan. One exists, the guide wire fixator, and the next one is the filter fixator. It's the same anchoring unit, but with a micropore filter added. The third product is microcatheter fixator, and it's the same anchoring unit with microcatheter fixated. The intended use is to facilitate endovascular treatment methods where temporary anchoring in peripheral artery is indicated. A ruptured aorta means normally immediate death. A normal aorta is 2 centimeters, and in most countries, all male over 65 are screened with ultrasound to detect if the aorta is growing. At 5.5 centimeters, an intervention is necessary, and you put, with endovascular technique, you put in a stent graft. That's been done since 1991 and standard treatment. In the complicated, 30% of the cases are complicated and can only be carried out in university hospitals. And the rest of patients will be left untreated or done with open surgery, which can lead complications and worse. The guidewire fixation will enable the county hospitals, which we already have the equipment for doing this, to do also the complicated aneurysms. Peripheral artery disease means narrow artery due to calcification and plaque, which lower the blood flow to organs and limbs. In the case of the legs, it can lead to critical limb ischemia with pain when the leg is at rest, and black toes, and widening of the blood vessel is necessary. The intervention is called ballooning or stenting or atherectomy. And during that process, the plaque and debris will flow downstream and block arteries further down. That's called embolism. And that can cause worse problems than actually the initial indication. So the filter fixator will be used in all intervention to protect from embolic risk in the legs. Microcatheter fixator can enable local drug delivery. The fixator secures a microcatheter in an artery near the organ, and an external pump infuses the drug to target over time. Catheter is stable when the patient is ambulant during treatment period, can be several weeks. Systemic side effects can be avoided, and the active substance can reach its target without first being metabolized. So the summary of the drivers: trend toward minimal invasive surgery, endovascular, better tools, more patient treated, prevalence increase with age in the addressed areas. Endovab has one technology and can access at least 3 large clinical fields. Products are single-use tools. They are not implants, and there are no active components, which is easier. The first product is commercially available for complicated aneurysms. The filter fixator is under development, and the microcatheter fixator is planned. This is the overall plan. We focus on verifying the market, growing sales, and build a company, and then expand geographically outside Europe. The next devices, filter fixator and microcatheter, they're not complicated technically, but regulatory-wise it will take some time. And to really hit the market, we need to do big clinical studies that is comparing standard treatment, and that will cost a lot of money and time in the future. Next step, commercial use have started. We aim for 100 patients next year and 1,000 the year after. Very conservative revenue next year, 500,000. Year after 3 million, and then hope to be basic operation profitable 2022. The coming devices are on plan. The key management is Chris Dewman, the inventor and founder, over 25 years of experience from vascular and endovascular surgery. I have long experience in management in life science, 10 years as production manager at Radi Medical Systems and St. Jude. It is endovascular too, similar to this, and 6 years as CEO of Nova Scientific, working with resorbable surgical mesh. In February, we'd like to get 15 to 20 million. Invitation will be sent out in December, info meetings in January, or book a separate meeting. We aim for the round 2 in 2022 for the major application-oriented clinical trials. You can contact me if you're interested. Be part of this exciting journey. Thank you.
Thank you so much. And next up we have Martin Vallet, the CEO of BrainCool.
Nice to meet you.
Nice to meet you too.
Okay, wonderful. So my name is Martin Vallet. I'm CEO of the Swedish medical device BrainCool. We started roughly 5 years ago with a lot of innovation spirits. We, of course, I didn't make this work. We have, with the aim to develop a product called BrainCool system, which is a surface cooling product in the growing temperature management market. Our aim was to make a non-inferior product to Arctic Sun, which is the project leader. This is a market where there's guidelines for cardiac arrest, guidelines for the neuro market, reimbursement in Korea, and a quite growing market. This treatment is really done on the ICU in hospitals for cardiac arrest and neuro patients. So the treatment window starts right now after 4 or 5 hours. So we complemented it with an acquisition of a medical device from the US called Rheumatoid, which has been assessed in cardiac arrest patient from the scene, right up from when you get cardiac arrest quite quickly, and then we would like to combine these two devices. We have started this company 5 years ago, so 3 guys with very high experience of the medical device business. So we got very good experience dealing with the FDA, CE marking, Japan, and business development. We run a number of clinical trials for our 2 products, are really in the launch phase now. Our product portfolio is the BrainCool system, which is then the surface cooling system. It's CE marked. We got Rheumatoid, which is CE marked. We're currently working on FDA approval, but we also developed products for oncology, even though the major business to do is brain cooling. I will tell you a little bit of our project in oncology last phase. We set up operation 2018 with the aim to launch our first product to the market, which is the BrainCool system, but under the brand name iCool system in the US. We've been very successful, not getting venture capital, but to raise money from the European Union. So we have 3 ongoing grants from the European Union funding efforts in stroke, migraine, and our oncology efforts. We are currently making a share issue, preemption rights, 40 million Swedish. We're listed on Spotlight, and that financial prospectus will be presented any day. This money will be used to fund the following. We're making a business plan in Asia. We got distributors in Korea, Vietnam has started sales. We're obviously setting up a goal to set up an own operation in the US. We have 6 employees, have launched our first product in April, got our first sales in August, and really working towards getting big in orders for the iCool system last quarter. Business plan in Europe is solely focused on the German market, the biggest medical device market, and sales penetration in Germany and the US will help us to build a further partner network. We are listed in Spotlight. We have enabled through Morgan Stanley in Boston to enable Americans to easy to buy shares in our company. The temperature management market, BrainCool system, Rheumatoid, is a hotbed for acquisitions in the US. All the other companies are built through acquisition. The most recent an Israeli product bought for $200 million last year. Most important, the temperature management market where these products are placed is really growing in the US. We have a market of $250 million in consumables every year for a product iCool system, and we expect to take a big part of that market. As cooling now is only done in hospitals, we expect through the product Rheumatoid, where recent trials will be presented at American Heart this year in 2 weeks' time, that the market will grow also in implementation in the EMS system. Rheumatoid, small portable cooling unit used with a catheter. A cooling agent is flushed up through the nose. You can cool for 2-3 hours with the aim to cool down the brain. It doesn't work for many hours, but when you come to the hospital, you connect to our other product, BrainCool system, where cooling could go on for neuro patients in the US up to 30 days. Other products is not approved more than 5 days, so we really are the continuum of care company. So the trick here is to, in the street, when you get a cardiac arrest, in the ambulance, start cool the patients as quickly as possible. Bam! Through the ambulance, through the emergency room, through the cath lab, neuro cath lab, cardiac cath lab, until you get to the ICU. ICU, that's where the other competing products are. And why should you cool early? All animal studies, all feasibility study, all our trials say the cooler the sooner you cool, the better results, which is also part of the American guidelines. So in the guidelines, they clearly state cool as quickly as possible. But there has not been any product available until our first mover product, RhinoShield system. So there are guidelines. A non-reimbursed market in the US, but a growing market because emergency medicine could pay for their own product. There are guidelines in Germany, the guidelines in Japan, the guidelines in Korea, which is the 4 markets we focus on. RhinoShield system, as I told you, there is no competition. On the other hand, it's a new clinical practice, so it takes time to deliver new clinical practice. We have trials for more than 1,200 patients and are whipping now to close partnerships to launch this product. We have very, very interesting development in Germany. So 9 university hospitals in Germany has applied for fast-track reimbursement for this technology in Germany. Read, there is no reimbursement in the high temperature management market. So this could be the first one that really could kick off. We sell RhinoShield for 600 to 1,000 euros per patient already now in Germany, and have had 2 cities launched this summer that decided to implement this on every patient in the ambulance. We have education in Halle, a small town in Germany, training 80 doctors, training nursing, training firefighters. We really have a good path. The BrainCool system faced severe competition. It's barred. Becton Dickinson, it's Sol, it's Stryker, it's GeneTerm, it's private equities that buy Israeli products. Tough competition. Let's see in the US what we can deliver. Talk is cheap. We think that only 5% of the potential market is cooled today in the US. That market is probably 250 million US, growing. In particular, Nevro market, where our products are extremely well fitted in terms of patent and technology. So current solution, I don't have a... You start cooling there after 3-4 hours with products like BrainCool system. Our pitch, you should be able to cool already after 10 minutes. We're working on integration of these 2 devices. We got a lot of EU funding for that and got very interesting coming trials also in stroke area. So hypothermia works, there's an induction phase, maintenance phase, and rewarming phase. And most products, with the exception of Arctic Sun and maybe a Thermogard from Sol, are not very precise. It's just water blankets. The BrainCool system, RhinoShield system, is a very, very precise engineered software-driven system with very, very good control. On top of that, if one like, we could kick in RhinoShield Future to really cool down the patient very, very quickly. This is a sub-analysis of the Princess trial, which was a clinical trial of the RhinoShield. I don't have time to go into detail, but the trend is clear. This is a comparison of the results if you start cooling after 25 minutes, 20, 24, 30 minutes, or even quicker. Read, the other products start the cooling after 4 hours. You get much, much better results, almost complete neurological recovery, 70% of the cardiac arrest patients. If we can transform this into the stroke area, you don't need any sexy biotech projects. Let's focus on the devices instead, which is strange in a town like Lund, where there hasn't been one pharmaceutical actually developed, but there is a number of medical device companies. Come on, somebody needs to speak up. But it's not me, because I'm too poor, as they told me here before. BrainCool today. We got 2 CE mark devices. Really, this is a company that's been up for 5 years. So 5 years. Look at the other medical devices. We got 2 CE mark devices. One of them FDA cleared in 2 510(k) approvals. We got approval in Korea. We have a very software-driven product, the first one, so you need to keep track on the software changes. That has taken us time to match to get this product approved in Japan. We're not far away now. We got 2 de novo 510(k)s ongoing with RhinoShield and an oncology product. On the oncology product, we have an expedited access pathway. No other Swedish medical device company has got that. I don't have much time for oncology today. We have a clinical trial of 180 patients with Karolinska. I'm on single digits. We completed product evaluations, industrialization, we got a lot of patents, reference customers, and the route to market is defined. I fear that I don't have so much time. Is that correct?
That is correct.
I just want to give you a last pitch. Remember, 70% of the patients from the Princess trial, full neurological recovery. We're doing trials now in Germany, 10 neuro ICUs. We have contacts, better than Sedona Medical. If we can prove that this works in stroke patients, complete neurological recovery in stroke, how much money do you save in Germany every year? 2.2 million euro next year. I don't get so many meetings in Stockholm, but Sol, Terumo, the big medical device companies, they work with me. Set a meeting with me, Fredrik, come on.
On that note, I think that's a perfect note to end on, isn't it?
But there's a guy here from Lundbeckfonden. Give me 10 seconds. Look at this device. Be quiet. We don't have any staff in oncology, but we know what we're doing. We developed an oncology device, 180 patients with Karolinska, prevention of oral mucositis, $40,000 per patient is the healthcare cost in the US. I got patents in 7 months and a fast track. You should speak to me. I don't have time. Marketing competition, blah, 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 you want to... I'm not the 10-minute presenter. And Stefan at Penn said, you don't want to meet me? I'm fine with that. But listen to this summary. Spotlight market. We're going to make the same journey as other companies. We're going to hit First North next year. We're going to be on the stock exchange 22. I accept that I haven't been able to raise any venture capital. But we are a very, very interesting company with 6,000 shareholders, but no real shareholder base. So I should have spent more time in Stockholm working on these foundations. But we are an extremely interesting company.
I think we're really going to have to give the stage to Jan now. Thank you so much.
If there is any real medical device industry here in the Medicon Village, let's make the pitch. The successful companies in the South Sweden is device companies, not biotech. Come on, give me a break. I can help you.
Well, thank you.
Sorry.
That is quite all right. But yeah, let's give the stage to the CEO of Follicum, Jan Alenfall.
No problem. Thank you very much.
Yeah. After
Hello,
what
everyone.
you do. Here you go.
Hello, everyone. So my name is Jan Alenfall. I'm the CEO of Follicum. So maybe this is the company from Lund that's going to make it and maybe do some good pharmaceutical products to the market. So we are working on peptides that come from our own body. The company was formed in Lund 2011, and we have at that point no experience in hair growth because that was the founder's idea that a modified version of an osteopontin protein could give rise to new hair. So 2012 we started to collaborate with international experts in hair growth, mainly from Germany and Israel. And in 2014 November we became listed on Aktiestorget, or today Spotlight. When we were investigating mode of action for the hair growth product, we stumbled across another interesting project which we founded in 2017, diabetes. And 2019 we developed our new topical formulation for the delivery of the peptide for hair growth. So the first project is hair growth, and hair loss market is a great market. It's a good, let's say, a very interesting market with a huge potential. So you normally think about male pattern hair loss, males in like my age, losing hair. A lot of people do that, causing a lot of trauma. But there is a lot of women also losing hair. And maybe that's more traumatic to them to lose hair than males. And this is a global market. It can be cultural differences between, for example, Europeans or Americans if you compare to Asians. So today there are only 2 products on the market. They are quite old. They suffer from a little bit side effects, and they are not efficacious. In the pipeline, there are not a majority of interesting products. There are a few. Some of them are in stable development, meaning that they haven't reached any, I guess, efficacy points. And today, most of these patients are left alone with only help from these products or shampoos or other supplements that suppose a promising value to them. And today, we only have finasteride or Propecia as a tablet. It was originally developed as a product for benign prostatic hyperplasia. It's a tablet prescription only for men. The other product is minoxidil or Rogaine. You can buy it in the pharmacy. You have to apply it 2 times daily. And altogether, these products have limited effect. And also the response rates are poor. Maybe 25 to 30% of the patients trying these products seem or appreciate the hair growth response. There are also some side effects that may limit the use of these products. And of course, only minoxidil or Rogaine is indicated for women, which limits their help, sort of. The market today is $3.5 billion for these 2 products, and it's about half in each product. And it's estimated in 4 to 5 years that it will be double. We have recently concluded a phase 2 trial with our product, and this product was injected 3 times weekly during 3 months on patients with hair loss. We can confirm that the safety is very good, no side effects. We could also see that we had an increased hair density with 7 hairs, although this wasn't statistically significant at the point. But also the other hair growth parameters, like the proportion of anagen hairs, that the number of hair follicles producing an active fiber increased, and the ratio between the fiber-producing hair follicles and the resting hair follicles was also found to be increased, which is very important. We also saw an increase in hair length and hair growth, daily hair growth, which is important. And for placebo, most of these parameters went in the other direction. So this table is just to compare our product with the marketed products. And you can see that minoxidil, the top row, after 6 months treatment with an optimal formulation twice daily gave rise to 7 hairs. And finasteride for 11 months treatment, also 7 hairs per square centimeter. And our product in our recent phase 2 trial gave rise to 7 hairs, but only after 3 months with only 3 weekly injections. So we hope with the new topical formulation we can really improve the efficacy and also the response rate of our product. So this graph shows you the comparison with the commercial minoxidil, 2 times daily application, in this case in a mouse model, and compared with our formulation once daily with FOL005 seems to be equally effective in this model. And we haven't gone up to the highest dose possible in this trial. So the next step for us is to induce a new phase 2 trial early next year, and we are preparing for that right now. We have had a scientific advice meeting with the German authorities, which was very good. Bridging tox studies cleared, no side effects, and now we are doing the product production. So basically, we want to achieve a safe product, easy to use, that can be used for all patients, both males and females, with a convenient formulation, once daily application only. And that would differ very much to the existing products. Now, the other product is diabetes. And you all know that diabetes is a big indication. More than 500 million people are today suffering from diabetes, leading to lots of complications. For example, beta cell loss of functionality, that the beta cell doesn't respond to high blood glucose and don't release insulin. So there is an unmet medical need in this indication, and we think we have a solution. Our peptide is actually distributed to pancreas. You can see that after subcutaneous injection, the peptide actually accumulates in pancreas. And the figure to the right, maybe not clear, but this is actually an islet, a Langerhans islet in the pancreas, and the red stuff is our peptide binding to the beta cells. This is another study, in vivo imaging system. This is the same mouse up to 48 hours, and we can follow by using a fluorescently labeled peptide that it accumulates into the center of the mouse, perhaps to the pancreas or the liver, but this is very interesting. The most promising thing is that we can actually protect beta cells. So this is an experiment where we induce glycotoxicity. So we expose cells to high levels of glucose that makes the cells sick or they become sort of toxic. And we have a number of peptides that actually was tested, and you can see one of them is GLP-1. Doesn't work very well to protect the cells from glucotoxicity, but you can see peptides from 6, 7, 8, 9, and 10 actually can induce beta cell protection and therefore recovery so they can respond again to high blood glucose levels. So this is really, really important. This is actually what happens when you have diabetes. So we have a number of benefits in our project. We think we can help patients with diabetes for their complications like cardiovascular effects, insulin resistance, and I mentioned protection of beta cells, anti-inflammatory effects, which is also very good in diabetic setting, and potentially also NASH. So what our molecule differs from, for example, GLP-1, is it's much smaller molecule, less cost of goods. It's not immunogenic. We know the receptors. It could potentially be first in class with a new target. And with the mode of action, we can actually also combine therapies, which could be important in the future. So how do we work? We are a virtual company. In the diabetes project, we are part of this big network, Lund University Diabetes Center, which was founded by Stiftelsen för strategisk forskning to up to 100 million Swedish crowns. So here we are part of several work packages. We have access to skilled people, we have access to models, and this is really, really interesting. So in this case, one postdoc is actually fully financed to elucidate mode of action, how these peptides work in for example, protecting beta cells. So that's really important. We have a small team, and with this, I thank you for your attention and leave you with a summary slide you can read. Thank you so much. Enjoy the rest of the day.
Thank you so much. That was really good. I think it's time for some more coffee, and I definitely need a cup of tea. So I will see you back here at 20 to 12. Thank you. think we will start. And our first speaker for this session is Monica Walter, the CEO of Leeds.
Thank you very much.
You're welcome.
So Leeds is a drug delivery, drug development company. We are based in Uppsala, Sweden, and we are having our headquarters at Uppsala Business Park. This company was founded 2003, and today we have a quite high number of different projects in different stages, from preclinical stage to phase 2B. So everything in our company is actually based on the nanosolid technology, a technology that is developed by Leeds, it's patented by Leeds, and we do have the possibility to actually tailor-make the release time of the actual drugs that we are formulating. So as you can see here, we are in different areas. We are focusing on oncology and immuno-oncology, and that's where our major work is continuing. So what's the benefits of having a drug delivery technology with sustained delivery? Well, of course, first of all, the technology is very friendly to the body, so it will be removed when the release of the drug is finished. Secondly, also, it can formulate most of the drugs that we've tried. So we have a quite broad spectra of molecules, from the really small ones up to polypeptides. So if we would like to have a product or a drug formulated for 1 month, 3 months, or 6 months, that's possible. And actually, I don't know if you've seen it, but today we announced a message to the market that in the prostate cancer study that we have a follow-up period with, we actually seem to have between 8 and 12 months of delivery of the anti-androgen drug for the Liproca product. So it looks really good for the nanosolid technology. This is just a picture showing how the product looks like. When it's injected into the tumor or the tumor area, it will be solidified, and it will be releasing the drug until the time that has been decided for this specific drug product. So for the patient, of course, there are a lot of advantages. You can get less injections. You can get less side effects, since you are not giving the drug directly into the blood. You are only giving it where the disease is. And that's a big advantage. That means quality of life improvement. And also for the pharmaceutical companies, this is something that can help them to actually tailor-make their specific molecules so they can act in a better way, and also giving the efficacy but still limiting the side effects quite substantially. So we have a very good patent situation. We do have patents for the technology and for prostate cancer. The technology as such, nanosolid, has patents in US and in Europe until 2037. So we are well protected. I don't think there is any pharmaceutical company having 18 years still of patent protection when developing drugs. So we can actually help pharmaceutical companies also to manage the life cycle of their actual APIs. Our business model is built on a traditional licensing model. So we expect to have collaborations and license agreements where we do get milestone payments and, of course, royalty. And having a long patent protection, as you know, that also counts for having quite substantial royalties during many years. But we could also have co-development agreements, and that will be the case in case we do work with a molecule that is not without patent. Then we need to, at least if we would like to enter phase 1, then we would like to have a collaboration because it's not possible otherwise. But generally, we would like to develop our drugs to phase 1 or phase 2. In certain cases, we could also out-license our projects in the preclinical stage. So we have a lot of triggers coming up, and some of these are actually already achieved. We did announce the preliminary data of the prostate cancer study just a few weeks ago, which shows that we have a persistent and good PSA reduction and that we have a very high response rate on the trial. I will revert back to a little bit more about that study. As you can see also, we are working with docetaxel, that's the DTX project, which is currently in phase 1. And the first patient is treated without problem, and we do foresee a few patients coming in later this autumn. Immuno-oncology has been a very, very important part of our development work lately. We do see the trend in the oncology field that immuno-oncology treatments are heading towards combination treatments, or maybe even triple or quadruple treatments. And we have found out a number of molecules that could actually be very beneficial for the technology and for the patients, because many of these drugs are giving very severe side effects given into the blood systemically. So we do have quite a few there, which are very intriguing and very exciting. So I will also talk a little bit more about that. This is actually our portfolio as it looks today. You see on the top, it's the prostate cancer drug, which is an anti-androgen drug, where we go for patients that has a non-aggressive cancer but still are not getting a treatment. And then the docetaxel, which is giving cytotoxic drugs directly into the tumor, where we're heading for head, neck, lung cancer, and a few others. And then we have the immuno-oncology molecules, which are not able to be given into the blood, like STING agonists and TLR9 agonists. So beside of this, we do have a few which we have chosen because they are so toxic that if you give them systemically, the patient will suffer a lot from it. So we choose the agents based on our technology and based on that these would not be able to be given otherwise. Well, this shows a little bit about the flow, how we plan to out-license the different projects we have in the company. So coming back then to the prostate cancer drug, which is the most advanced project at LIDS, we have just finalized the phase 2b trial, and we have done a number of phase 2 trials before, and they all say the same. We already have a license agreement with China. Poheng Pharma is currently working with the phase 3 synopsis and preparing for having the discussions with the SFDA in China. So, of course, in parallel, there are ongoing discussions and work to get agreements signed for the rest of the world, US and Europe. So which group are we heading to? As you know, prostate cancer is a disease that has a lot of different stages. And as you can see on this picture, we are going for the green part, not the ones that has a very low risk, not the one that has a very aggressive cancer that should have surgery or radiation very soon after the diagnosis. So we have found a patient group that corresponds to approximately 420,000 patients a year, which are eligible for a leproca treatment. And knowing that the amount of prostate cancer diagnosed patients is approximately 1.2, 1.3 million a year. So it's approximately 35% of the population. So I won't go into the detail of prostate cancer, but it's very good to find out in the preliminary results that we have a very good result in this target group that is actually the group that we have worked to help. So this is very favorable for LIDS. It goes directly into our TPP, the target product profile. The study was built up in 2 phases. The second part was more efficacy, whereas the first part was safety, because we actually would like to see the increased dosage that it worked safety-wise. It's also interesting to note that we have a follow-up injection for certain part of patients. And as I said, it's approximately 8 to 12 months efficacy time. And that goes directly into the system that we can actually treat these patients once a year, and hopefully then they can avoid surgery and radiation, which is having a lot of side effects like impotency and urinary incontinence, etc. So, yeah, we are very happy with the phase 2b results. We are very happy about the open label study that we just released data today. And if there are any questions for the other projects, I'm very happy to respond to this after the presentation here.
Thank you so much, Monica.
Thank you.
Then we will hear from Scintilla, I think. Welcome up.
You.
Ja, du har en egen. Stänga av den där.
Okay, thank you very much. So my name is Thomas Askog. I'm a BD manager at Scintilla, replacing our CEO Eva Lundgren-Åkerlund today. So we're a Swedish biomedical company located here at Medicon Village, actually, a great place to be. We're a team of 25 people today, approximately, and we've been listed at Nasdaq First North in Stockholm since March 2016. So we develop products in two main areas. So that's cell therapy and in oncology, where we focus on indications with a high unmet medical need. So our current focus in cell therapy is on the degenerative joint disease, osteoarthritis, and in oncology, the brain tumor glioblastoma. And although these are two very different indications, we actually base our product development on the same proprietary integrin marker technology platform. So the marker that we focus most of our work on is an integrin called alpha-10 beta-1 that initially was discovered as a specific marker on chondrocytes or cartilage cells. Later research showed that certain mesenchymal stem cells also express our integrin marker, and that it is strongly upregulated in certain cancer cells, and this is what we utilize in our product development. So over the years, Scintilla has developed antibodies that bind to the integrin marker that we use in our product development, and we use them in two main ways. So in cell therapy, we use them to identify and select high-quality therapeutic stem cells, or mesenchymal stem cells, MSCs. And in oncology, we use them to target aggressive cancer cells with a drug. So in cell therapy, our first focus is on osteoarthritis, which is a degenerative joint disease with a degradation of cartilage in the joint over time. It's a huge problem affecting over 600 million people globally, and it's a problem that increases with age. And the global market for OA drugs is huge, and it's growing rapidly. And of course, we want to take a part of that. So we develop what we call an off-the-shelf mesenchymal stem cell product for the treatment of OA, both in humans and in animals. So we use what we call allogeneic or donated stem cells from adipose tissue. So with that, you can use stem cells from one donor to treat many patients. So it's a very cost-effective approach. And we're also aiming for a simple one-step procedure where the stem cells will be injected directly into the OA joint, so you won't need any surgery of any sort. So what makes us unique? Of course, we have competitors out there developing stem cell products for OA and other indications. What makes us unique? So this is explained here on this slide. When you isolate stem cells from adipose tissue or bone marrow or other sources, what you really get is a heterogeneous mixture of cells. So in this mixture, you have some true stem cells, but you also have progenitors and other cell types. And this preparation is what most other companies are using in the product development, and that causes a lot of issues, not the least from a regulatory point of view, because problems to show product identity and consistency. So we solve this problem by selecting high-quality stem cells based on our marker to obtain a homogeneous stem cell population that we then use in our product development. So with this, we get a homogeneous and consistent stem cell preparation. And that brings a set of advantages, for example, functional advantages. We know that our selected stem cells, for example, have a much higher chondrogenic differentiation capacity, so they have a high capacity to differentiate into chondrocytes, as shown here in a pellet mouse assay. So these are selected stem cells. So shown here by staining or collagen type 2, which is found in high-quality cartilage. So in addition, we know that our selected stem cells are immunomodulatory, anti-inflammatory, and that they also have an increased ability to adhere or home to damaged cartilage. And this is really important. So they can find the damage and to the place where they should exert their function. We also have several regulatory advantages with our approach. So as I said, we get homogeneous cell preparations. We get consistent quality between donors and batches. We can use our market technology as an in-process control and quality control, and to predict the potency of cells, which hopefully will show on the clinical outcome. So we get a defined and consistent stem cell product. We have analyzed our stem cells in a post-traumatic OA model in horses, where equine stem cells were isolated from equine adipose tissue, selected with our market technology and frozen down in vials, shipped to our collaborator at Cornell University, New York, Dr. Lisa Forcier. So they injected our stem cells in one damaged ankle joint, and in the other got a control, a saline solution. And after 6 months, the cartilage, underlying bone, and other tissues were analyzed. This is some results. If you look at the cartilage, so this is a cartilage from a non-impacted area, stained here with Cephanin OA, Cephanin O, showing a really nice cartilage. This is in the control. Here you can see that in the impacted area, no cartilage at all. You can see a lot of fissures. And this is in the treated joint, much better integrity of the cartilage. And this was a quite small study, 7 animals. It was statistically significant between the groups if you looked at cartilage fibrillation, so less cartilage fibrillation in the treated joints. And also a much better result also when it comes to bone sclerosis, which is another important hallmark for OA. So in the group treated with the joint treated with our MSCs had a better sclerosis score and also a thinner subchondral plate. So we reached several important milestones, positive preclinical results. We now have a scaled-up production of stem cells in the bioreactor. And importantly, we have built our own GMP facility for production of stem cells for our upcoming clinical trials. And this is, I think, very important because it builds a lot of value in the company, because as a partner, we do not only bring a unique stem cell platform, but also the manufacturing capabilities. We're preparing for our first in-human study. We have a clinical study protocol in place, and we are finalizing negotiations with CRO and study sites. And we recently had a scientific advice meeting with the MPA regarding the clinical study, and with very positive response. So the next milestones, we are waiting for an approval for the GMP facility. We're going to initiate our first in-human study with our first product, XTEM-OA, for treatment of knee OA. Initiate co-development with a partner for OA therapy in animals, and also do more preclinical evaluation for new indications for stem cell treatment in the orthopedic and possibly CNS indications. A few words also on our oncology project. Here we develop antibody-based products for treatment of glioblastoma as the first indication. Glioblastoma, as you know, is the most common and most aggressive form of brain tumor in adults, affecting some 25,000 to 30,000 individuals per year in Europe and the US. And also here we have a substantial global market for glioblastoma treatments. What we develop is an antibody-drug conjugate. It's a first-in-class approach. We see that our integrin target is strongly upregulated in glioblastoma tumors. So the idea here is to target the cancer cells with an ADC that delivers a toxins that kills the cells, the glioblastoma cells. So we have several positive preclinical results. I don't have time to go into the details here, but we have shown that the ADC binds to glioblastoma cells and has a killing effect both in cell studies and in animal model. So we have a therapeutic target with large potential in oncology. Positive preclinical results that were recently published in a distinguished journal called Cancers. We have identified new cancer indications recently to focus on, and we have filed a new patent applications for diagnosis and therapy of aggressive cancers based on our technology. So the near-term milestones here is to identify antibody candidates, to do proof of principle studies in animal models, that's ongoing, and to evaluate and decide on new oncology indications in addition to glioblastoma to focus on, and to identify partners and licensees. What we have also done is that we have spun out our oncology business to a new company called Targinta. So Targinta will focus on cancer therapy, the products, because it's developed so well over the last 2 years. And all the stem cell therapy products will remain in Xintela. So we're looking for partners and investors for both companies. So final slide, why Xintela? We do have a patented market technology platform with large diagnostic and therapeutic potential, stem cell therapy expertise and technology with unique advantages, and importantly, in-house GMP manufacturing facility for the stem cells. We have animal health projects with shorter time to market. We have a first-in-class oncology target for therapeutic product development and extensive IP covering products, methods, and use claims. Thank you very much.
Thank you very much. And then we welcome the CEO of Omnio, Patrik Stavsede.
Thank you. As mentioned, my name is Patrik Stavsede and I'm the CEO of Omnio. That's a spin out of Umeå University up in northern Sweden. And I'm going to be a bit of a contrarian today because you listened to Cereno's scientific and you heard how important it is with anti-inflammatory properties, as well as Aptahem, who tells you what happens when an inflammation goes haywire into a sepsis. And I'm going to talk about pro-inflammatory drug development because that's where we're a pioneer. You all know that inflammation is the way that the body responds to trauma or to injuries, as we call it. It could be an infection with bacteria, it could be a trauma where you hit yourself on the thumb, or it could be just an incision wound. And all these responses will generate inflammation. And it will be heat, it will be redness, swelling, and that's because the inflammation is going a little bit too far. And that's when you get pain and you get fever, and you need to reduce that with anti-inflammatory drugs. And those are the most common ones that we have. We have aspirin, acetaminophen, diclofenac, and naproxen. But where do the pro-inflammatory drugs come in? What do we do if the body just doesn't respond as it should, if it doesn't give you the inflammation? There is a professor up in Umeå University who has been working with plasminogen for 30 years. He is Professor Tourny. He founded the company, and he discovered that the protein plasminogen actually regulates wound healing in all of its different phases. Because when it goes to wound healing, it's 3 phases. It's first an inflammatory phase, then it's a proliferation phase, and a remodeling phase. And this protein plasminogen is quickly concentrated where the area of the injury happens. So plasminogen is a protease that is produced in the liver, so you have it in all your bodily fluids on a low concentration. But when it's needed, it's rapidly concentrated to the area of the injury. But what happens is that plasminogen is a key regulatory molecule, so it takes care of all the time span that needed from the initial injury until the wound is entirely healed. So the injury will activate the plasminogen receptors on the surface of the cells. Plasminogen binds there and it's activated by TPA or UPA, and then it leads to a lot of different processes in the body. It regulates gene expression. Some are upregulated, some are downregulated. It removes bacteria, it removes necrotic tissue, and it can help heal several different indications. So what we have been working on is wound healing as the number one, both for the worst ones you can think about, diabetic foot ulcers, but also chronic perforations on the tympanic membrane. But it's also other types of wound healing that you don't consider wounds immediately, like periodontitis. And it can also help against infections, help the host defense system. So, plasminogen. It can actually stimulate wound healing where there has been a stalled chronic wound. So these pictures aren't nice, but there are patients out there for where the amputation is the only other alternative, and they're willing to test things. And here we have a patient that had a chronic wound for 3 years that he couldn't walk on. But after treatment with plasminogen, it took about 3 weeks with injections every 2 days. And as you can see in the middle picture here, the wounds are healed and you can start walking on your feet again. So this creates a tremendous benefit for the patients suffering from this. The alternatives that we mentioned for diabetic wounds is often amputations of lower limbs. And these amputation wounds tend to have a bad healing as well. But here we can inject locally plasminogen around the injury. And it will stimulate the body's own mechanism to heal the wounds. So what's good is that we're having a biological feature that then shows no side effects because it's already in the body. We're just concentrating it to the spot on the body where it makes most benefits. And of course, these diabetic wounds, they cause a huge financial burden because of the 300 million diabetic patients, about 10 to 20% of them will actually develop diabetic wounds. And there are millions of amputations ongoing in the developed world every year that could be avoided with a simple biological product. I also mentioned chronic tympanic membrane perforations. Today, the alternative is that you actually cut open your outer ear and you flap it forward so that the surgeons can access your ear canal and put in a little thin paper, which they hope will integrate with the membrane that's perforated. With the plasminogen injections locally, we can stimulate the tympanic membrane to heal by itself. Then, as I mentioned, periodontitis is not what you really think about as a wound healing, but we see the same process there. And periodontitis, that's a very common disease. We're talking about 700 million people in the world having severe periodontitis that will lead to tooth loss. And you all know that gingivitis is very common. And that is one stage of the disease, but it's not dangerous because it doesn't cause the bone around the tooth to break down. But if you're looking at the periodontitis on the right hand, you can see that there are gum tissues falling apart from the tooth, and there is a deep gum pocket, and the bone will actually start to break down, which leads to tooth loss. But when we treat mice, it might be hard to see here, but when you have the tooth here on both sides, you have healthy gum tissue, and you have an alveolar bone in between that's healthy on a healthy mouse. When you're looking at mice that don't have any plasminogen, the bone will break down and the gum will be very distorted. But when we give them plasminogen, not only can we stop the periodontitis, we see that the alveolar bone actually regrows. And that's what you see over here. And that's what makes dentists enthusiastic when we talk to them. They said, wow, can you actually regrow bone so we have something to put the dental implants in if we need to restore that? And can you stop the tooth loss? If this is transferable from animal to humans, that's something that's going to revolutionize the dental implant world. We have also shown that plasminogen can actually treat bacterial infections. So we have shown that in mice, where we have infections with MRSA in the knee joints, we can actually lower the bacterial count when we treat it with plasminogen. And the same with E. coli that we've just done recently. We can show that 12 days into the treatment, we can reduce the bacterial count with 100-fold. And if we can solve this, then of course, the benefit is that there is no antibiotic-resistant development because we're helping the host defense system of the body to be engaged instead of killing the bacteria. So we have patents for all of these 4 indications that I mentioned: the chronic wounds, the tympanic membrane, the host defense against infection, and the healing of periodontitis and periimplantitis. And we also have an ongoing patent file for reducing the side effects from radiotherapy, which is a different subject I can talk to you later about. But so far, the first 2 indications are out-licensed. There is a phase 1 that is currently paused at the Karolinska, where they're trying to do tympanic membranes. We have an approval from the MPA to start the diabetic foot ulcer treatment here in Malmö and Lund. And now we are focusing at Omnio on the preclinical development of the other 2 indications that we have. So if you want to join us on this journey, I'm very happy to take questions after the break.
Thank you so much. And then we will have our last presenter before lunch, Imre Zeilermacher from Immunovia.
Thank you. I'm here to tell you something about Immunovia. My name is Simon Zeilermacher. I'm the Director of Business Development for the company. We're public on the Nasdaq Stockholm. We went IPO actually in 2013 to the Nasdaq First North, but beginning of last year we went to the main market. We are focusing on bringing a unique cell-based immunotherapy for solid tumors forward in clinical development. We just completed a phase 2 in kidney cancer, and I'll show you some of the results we have. And in different studies, we've now completed already treatment for over 90 patients with a very good safety profile. Our manufacturing is taking place in Germany. It's an allogeneic, off-the-shelf cell therapy manufacturing, but we're also transferring that to the US to a site in New Jersey to really be ready for a commercial scale of our development. Our studies have been mainly performed in Sweden, then expanded into Europe, and now we have studies also ongoing in US. So there's a lot of regulatory experience now around the program. And beyond that, we also have a collaboration now in place with Pfizer and Merck Cereno. That's purely for a collaboration of supplying their checkpoint inhibitor for the new study that we're conducting. But that also validates our program from that area. As I said, we're public at the Nasdaq Stockholm. The last financing we did was in December 2018, around $36 million, 350 kroner, and that really helps us to continue our development for the coming 12 to 18 months and generate more data that supports our program. We have a small team of about 16 people, mostly based out of Stockholm now, but we do have all the in-house expertise in terms of regulatory development, manufacturing, and of course, important for the future also, the pharma and business development side. So what are we developing? As I said, it's an allogeneic off-the-shelf program, which means that we start from a healthy donor, we take out immune cells, and we activate these immune cells to become inflammatory dendritic cells. Now, we're not using these as a vaccine, but we're purely using them for an inflammatory function. So when we inject these cells into the tumor, you will create a personalized immune response against that tumor tissue of a patient, while the product is always the same. It's always an off-the-shelf product coming from a donor. There's no matching needed between donor and patient. And from one healthy donor, we get about 100 doses, and we inject patients only twice. So we can treat 50 patients from any blood donor. And because we cryopreserve, we freeze the material, we already have a 3-year shelf life. So for a cell therapy, this makes it actually a very straightforward way of manufacturing it. Now, after we inject these cells into the tumor tissue of a patient, and that can be the primary tumor, but also a metastatic lesion, we create this immune response against the tissue. So how do we do that? Our cells are purely producing inflammatory molecules called chemokines and cytokines that are there to warn the immune system that something is going wrong. So the tumor is very good in evading the immune response, but because we're creating this, let's say, local bomb inside the tumor, we're warning the immune system to come in and see what is happening. So then we get actually the immune cells of the patients that are doing the work, starting to attack the tumor cells, recognize the tumor cells, and in the end activate the patient's own killer T cells, as they are called, as the end product of this mechanism. So only 2 injections of our therapy, and the end product is a more systemic T cell response that can help not only this tumor site, but also metastatic lesions throughout the body. Now, we've tested this program already now in 4 different clinical studies. We started in kidney cancer because in kidney cancer we had the opportunity to inject into the kidney cancer while following also the response against the metastatic lesions. That was a very small study in 12 patients in Sweden that we started in 2012, so quite some years ago. But because of that, we also have a lot of follow-up data of these patients that show the extended survival that you can accomplish in this indication. Now, after we completed the phase 1, we initiated 2 other small studies, one in liver cancer, 18 patients, also in Sweden, that was very promising. It's a very high unmet need in liver cancer, but we were able to show that an immunotherapy can make a difference there. We also had a very small study, 6 patients in a rare indication called gastrointestinal stromal tumors, which even though it was only 6 patients, we were able to show that this tumor is responding to an immunotherapy. More importantly, recently we received the results of our phase 2. So this was a phase 2 controlled study, meaning we had our immunotherapy followed by the standard of care, being sunitinib, versus sunitinib alone. So 58 patients in the combination study, and then 30 patients that only received the standard of care after the primary kidney tumor was removed. So we looked at response and we looked at survival and a few other endpoints. Now, looking at response, we know that sunitinib on its own already had quite a good response. However, patients quickly progress, usually in this indication. What we noticed on top of sunitinib is that we actually have more patients going into a complete tumor response, meaning there's no metastatic disease left for that patient. So we had 11% in our group versus only 1 or 4% in the control group. And we also saw the same type of signal happening for the other responses. So we saw that there was a longer duration of response, meaning that patients have a more stable response on the treatment and don't progress as quickly as they do on the control group. We also had much more responders that were still ongoing at the end of the study, and these responders were also alive. This is all related to what immunotherapies have been accomplishing in other indications, that if you can make this response more durable and deeper, as they call it, you also expect a better survival in the end. Now, regarding survival, we had the initial follow-up of only 18 months, but of course we have already treated patients that started 4 years ago. So we have that data as well. So after 18 months, there was no difference visible between both groups. However, if you look beyond the 18 months, you saw that actually in the combination group, a lot more patients were alive as compared to the control group. Now, this is something that we, of course, have to follow. We haven't reached the median overall survival, the average survival in both groups, but we will continue the survival follow-up every 6 months to see when we will reach this average and see if we see a survival benefit. But based on all the individual patients we're now following, we're already starting to see quite an interesting trend. Now, the next follow-up is going to be in January. So then we have at least 24 months survival for each patient, but also patients that started quite some years ago. We're going to publish that in January to the general market. And beyond that, of course, we're now doing a lot of preparations in terms of the next study. Because it was a phase 2 controlled study, we do expect this data to be sufficient to move into pivotal studies for kidney cancer. The market has changed since. We've seen introduction of checkpoint inhibitors in this indication. So now we have to go to authorities and discuss, okay, what is going to be the combination that we can move forward. Now, regarding checkpoint inhibitors, we are trying to position ourselves in combination with different classes because we only have 2 injections, because we have a complementary mechanism and a good safety. We will try out different combinations. And checkpoint inhibitors is a very logical one. It's a combination where we activate the immune system and they are able to block the defense of the tumor against the immune system. So this is a study that we started earlier this year. We now completed the first cohort. The first 6 patients we have to treat one by one for safety purposes. But luckily in the first 3 patients we didn't see any issues in terms of safety. And now we've moved to the next dosing cohort, the 10 million cells per injection, which is the dosing that we have used in the other studies. After the first 6 patients have been completed, we are able to open up the different cohorts. So then recruitment should go a lot faster and we can complete the phase 1. After we complete the phase 1, that's when we move into the phase 2, where we have the collaboration with Pfizer and Merck Serono. For the phase 1, we focus on using pembrolizumab or Keytruda, but for the next one, we're going to combine with avelumab or Bavencio. So as a summary, we have a very unique product in development in the immuno-oncology space. We're already quite advanced in terms of the clinical data that we have, and especially the safety that we have in different indications. We have the right team in place to really move this to the next stage of development. And regarding next stage of development, we have financing in place to prepare for that, and we are now exploring for options to move that into the right pivotal study. So if there's any questions, I'm happy to answer them outside and, of course, over lunch as well. Thank you.
All right. Thank you very much, Simen, for that interesting presentation. So I'm just going to say that now we are going to take 1 hour lunch break. And the lunch will be served at the Inspira restaurant, which is down the hall and to the left. And we should have a secluded area for all of you here. So, and the food will be served as you come there. So if you have any special dietary requests that have not been reported, you should just tell the staff, serving staff, and they will help you out with that. So we're going to start over again at 1:20 with the panel session. That's going to be investing in innovations from discovery to market, and we have distinguished panel members are from Swedbank Robur Medica, from Arctic Aurora Life Science, and from Danske Bank Asset Management. So be sure to be back at 1:20, and you don't want to miss that. Thank you very much.